The baculovirus is commonly used as a DNA vector used in combination with insect cell lines for protein expression. However, the fact that this virus efficiently infects insects with lethal consequences also makes it an effective insecticidal agent. In Brazil, for example, wild-type baculovirus is applied to more than 1 million hectares of soybean crops annually for control of the velvet bean caterpillar. To enhance their insecticidal efficacy, baculoviruses have been genetically engineered with genes encoding insect-specific toxins that are active within the hemocoel of the insect. In this video, we demonstrate methods for oral infection of Lepidopteran larvae with baculovirus in order to analyze insecticidal efficacy.
Hi, I'm Huang Li from Brownies Barnes Laboratory in the Department of Entomology at Iowa State University.
And I'm Wendy Sparks, also of the Bonding Lab. Today we will show you two procedures for baculovirus infection of Lepidopteran larvae. First, Huang will show you how to infect neonate larvae using a droplet feeding bioassay, and then I will show you how to infect mid to late instar larvae using a diet plug bioassay.
So let's get started.
Droplet feeding bioassays can be used for the determination of the lethal dose of a baculovirus and survival time of baculovirus-infected larvae. Early instar or neonate larvae are generally used for this assay. Before starting, make sure you have diet cups with the appropriate diet on hand. Also, prepare the appropriate virus dilutions. Include green food coloring dye to a 4% concentration by volume. In order to determine the lethal viral concentration, the dilutions should give a range of mortality between 1 to 99%. Preliminary bioassays may be required to determine an appropriate range of virus concentrations. After vortexing the tube to prevent settling of the polyhedra, make 2 concentric circles of small droplets of the virus solution in a 60 millimeter Petri dish for each concentration of virus used. Remember to also set up a negative control mock infection treatment. Next, transfer 25 to 30 neonate larvae to the middle of the concentric circles using a sterile paintbrush. Place the lid on the dish and seal with parafilm to prevent the larvae from escaping. Ideally, use different paintbrushes for the transfer to each virus type to prevent contamination between treatments. Allow the dish to stand at room temperature for 10 to 15 minutes, during which time larvae drink the solution. Larvae that have fed can be identified by green coloration of the anterior gut, resulting from the green food coloring dye. Transfer only those larvae that have fed to 1 ounce plastic cups, which contain diet. Transfer 1 larva per cup. After transferring the larvae, cover the cups with lids and incubate at 28 degrees Celsius. 24 hours after infection, eliminate any cups containing dead larvae. These early deaths likely result from handling injury. When using this bioassay to determine lethal dose, the larvae should be checked every 2 to 3 days until the mock-infected larvae and any surviving virus-treated larvae have pupated or have begun to burrow in order to pupate. Virus-infected larvae will remain on the top of the food and then die. After a few days, dead larvae will sometimes melanize and turn black. Larvae that die from infection essentially become bags of polyhedra, which will completely dissolve if mechanically disturbed. At this time, score the larval mortality in each treatment. Lethal concentration bioassays should be replicated 3 or 4 times on separate occasions. Use 30 individuals per dose for each virus or control treatment. Use an appropriate probit analysis program such as POLO or SAS to calculate LC50 values, 95% confidence intervals, and the appropriate statistics. This droplet feeding assay can also be used to determine ST50 survival time values. In this case, Larvae mortality is scored every 4 to 8 hours, with more frequent observations if the rate of mortality is high. Survival time bioassays are performed using an LC99 dose of virus. Median survival times and the 95% confidence limits are calculated using the Kaplan-Meier estimator using an S+ or SAS program.
Now that Harang has shown you the droplet feeding bioassay, I'll show you the diet plug bioassay.
In diet plug bioassay, mid to late instar larvae are fed on a diet plug treated with a known dose of virus to determine the lethal dose, or LD50. On the day prior to conducting this assay, remove the diet from the cup to starve larvae overnight. This step will increase the speed with which the larvae consume the virus-inoculated diet cube. The next day, prepare the diet by cutting it into small cubes, about 2 cubic millimeters. If the diet cubes are too small, they will dry out. If the diet cubes are too large, the larvae will not be able to consume the entire diet cube overnight. Now add 1 to 2 microliters of polyhedra suspension to each diet cube. The suspension contains 4% by volume food coloring dye. Let the suspension soak into the diet for 5 to 15 minutes. Remember to also prepare a mock virus-treated diet cube as a negative control. Next, transfer 1 diet cube to each 4th instar larvae in each cup. Then place cups in the incubator overnight at 28 degrees Celsius. The next day, discard any larvae that have not completely eaten the diet cube. Then add a large piece of diet to each cup for the remaining larvae. Maintain larvae at 28 degrees Celsius, adding additional diet as necessary until the mock-infected larvae and the surviving virus-infected larvae have pupated or begun to pupate. At this time, record the numbers of dead and surviving insects.
We've just shown you 2 methods for infecting insects with baculovirus: droplet feeding bioassay and the diet plug bioassays.
When doing this procedure, it's important to remember that the cadavers of baculovirus-killed larvae are typically fragile and can easily rupture, releasing virus. So sterilize work surfaces and dispose of contaminated gloves and disposable materials in an appropriate manner to prevent contamination. So that's it. Thanks for watching, and good luck with your experiments.